Please excuse me that I'm based in Switzerland, and I could not resist in taking this analogy to Swiss Army knives. I assume many of you know what this is. What you see. I have no shares in that company, but it's one of the two big companies in Switzerland. So it is a tool that has many functions, one for every perceivable need, and of course there are many other types of knives like this. But it is interesting that the term Swiss Army knife has entered popular culture as a metaphor for usefulness and adaptability. And I thought, well, that is a nice starting point. I will get back to the Swiss Army knives towards the end of my presentation. But we have seen throughout the day various definitions of adaptive designs, and the first few slides are to some extent repetitive because I very much agree what I have heard so far. So I'm going to skip essentially or browse through very quickly. And adaptive designs has to be prospectively planned based on accumulating outcome data from patients in a trial. Note that I have skipped integrity and validity because these will be part of the principles I'm going to show in a couple of slides. So they are captured by these principles. Why conduct adaptive designs? We heard that ethical, efficiency, and maybe also more general scientific values as well, because if I take a second dose into a phase 3 trial, then I get better understanding of the dose-response relationship, and I could select one of the two doses, but it is an incentive to take a second dose into a phase 3, which otherwise I might not do. Common types of adaptations, we heard them also throughout the day: early trial stopping, sample size adaptations, population selection, and treatment selection. So these are the types of adaptations that I frequently see in my environment. Note that I talk about types of adaptations rather than adaptive designs. A subtle difference. To me, it is an important difference. But yes, so we have heard about this before. So these are the principles that I briefly mentioned. And you see 5 principles. 4 of them actually come from an FDA guideline on adaptive designs in 2019. I do understand that in the session after the break, Greg Levin from FDA will be presenting possibly about these principles. So I'm not going to any details, just to explain that the very first one, adequacy within the development program, is something that I added because personally I see it's very important that we do not run an adaptive designs for the sake of innovation or for the sake of having an adaptive design. It has to fit, it has to be fit for purpose and fit my overarching development program goals. Now, these principles, of course, they would also encapture the validity and integrity that we have heard this morning. I think, Thomas, in your definition, you had integrity and validity, and I see them captured by this list. These principles would be relevant across all phases of clinical development, be it exploratory or confirmatory trial, although the practical considerations and implementation may differ. Importantly, I feel that having such principles in place could greatly help clinical teams plan and implement adaptive designs. It's almost like a blueprint. It really helps, like, okay, have I considered these 5 bullet points that you see on the slide? And I do ask sometimes internally the teams, have you looked into these? So for the rest of this presentation, I would like to focus specifically on reliability of estimation. And Philip Parman already in the morning provided, very nice overview. So setting the scene. And I will primarily talk about estimation in a confirmatory context. So when it comes to submissions, regulatory approval, and drug labeling. So what is the problem? Well, adaptive designs involve selective inference based on accumulating data. Selective inference is a fancy but established term in certain communities to indicate that we perform our inference based on some selection or decisions after having observed data. So that's what they call selective inference. And of course, adaptive designs will involve selective inference using accumulated data. And it is well known that that selective inference increases the chances of incorrectly concluding compelling treatment effects. Now, this can take different forms, these incorrectly concluding compelling treatment effects. You see 3 of them listed here. Increased probabilities of false positive conclusions, often under the heading of type 1 error rate inflation. Exaggerated treatment effect estimates. We heard them under the heading of bias throughout the day. but also confidence intervals may not have correct coverage probabilities. So the first bullet point about false positive conclusions, the increased probabilities, this is something we're all aware of. Most of our trials will be very well designed, keeping type 1 error rate in control. So this is in the front of the minds for many of us. But the other 2 points are surprisingly under-discussed or not discussed well enough when it comes to bias and with coverage and the properties of confidence intervals. And let me try to explain this with one sort of simulation. So what I did, I simulated 10,000 replicated pairs of study. In the one study, in each pair, the one study, I would just compare drug versus standard of care or placebo in one population, no selection taking place. And I would then plot the effect size on the y-axis, that is the vertical axis. In the other study that I simulated, I would then have still drug versus standard of care, but I would do that across 4 populations, independent patient populations, think about different age classes, and I would select the population with the largest effect size. And this is what I would plot here on the x-axis. And so I would do that 10,000 times. And this is the sort of scatter plot that you get. So each of the dots is one of these simulated pairs of studies. And then I have also included a green line. The green line is if you have no variability and if you have no bias, then this entire cloud would condense to a point on that line. More specifically, because I have run the simulations, I know the ground truth. And I have assumed that the effect of drug versus standard of care is one unit of something. So you can see that, yes, so this I have simulated. So this is the one effect across all 4 populations, and this is the one effect in the trial without any selection. Now, of course, there will be some variability, you know that. So I assume there will be a cloud, and I can expect that. But you see that the cloud of points is not centered around this triangle, but it is shifted. And it is completely horizontally shifted. And this has 2 messages. First, the cloud is pretty much symmetric around this one line for the studies without selection. So I have no bias. I can estimate with some variability my treatment effect in that sort of studies without selection. However, you see the horizontal shift, and this is exactly the bias that I'm observing if I select the best out of 4 populations. And I can quantify it. It's 1.2 instead of 1. So the practical consequence is that on average, I'm reporting a treatment effect of 1.2 units rather than 1. So it's a 20% increased effect size that I'm reporting. So reporting conventional effect estimates and confidence intervals. And this is what currently in my domain, in my environment, we are currently doing all the time, fails to account for selective inference and may lead to unreliable estimates and perhaps even to misleading conclusions. This is the problem. So that is why it is important to talk about reliability of estimation, although we don't do that enough. It is the least appreciated among the 5 principles that I had listed on the previous slide. When I talk to my teams, this is the one that they think least. And this is ironic because there's an extensive literature over at least 40 years on statistical methodology and related software implementations. And I'm so glad I'm here in the room with David Robertson and many colleagues that have just published a series of 4 papers just on this topic. It's really the state of the art. Just look into this. Now, you can correct me, David, but I think there's no consensus on preferred methods. And certainly from a practical side, there's very limited uptake in practice of these methods, although it's out there since 40 years. Now, which is not ironic, but it's really sad, I think estimation, this is critical for communicating with patients, physicians, prescribers. It's not sufficient to say, okay, the drug is approved, it is on the market, but I need to somehow communicate these effects. And I think this communication bit is really critical, but it seems that we are not yet doing a good enough job. And this is not specific to adaptive trial designs. I will have an example with me in a few slides, which is in a non-adaptive design, just to start my discussions, but it's not specific to adaptive trial designs. It's a general problem because this selection also can occur in non-adaptive settings. So what does then reliability mean? Well, blue sky thinking, I would like at the design stage of a phase 3 program, have a joint understanding of what the drug label looks like, and then with estimates, so the numerical quantities to be filled once approval is granted. That would be my blue sky thinking. So I would like to focus in my presentation on the what, not on the how. I don't want to talk about methodologies at all. I want to understand what should be reported. And I have two questions. The first question is from a statistical point of view. What are statistical properties, I call them desiderata, for point estimates and measures of uncertainty such as confidence intervals? What desiderata should be considered considering the trade-off between bias and variance? We should always be mindful it's bias and variance, and often they're coupled. Now you will, on the next few slides, see that I have negative news, that desiderata are available, but they are universally not achievable. Which then that raises the question, what information should then appear in a drug label considering the limitations of current statistical methodologies? That's the outline for the rest of my presentation. So let me first focus on statistical desiderata. And the very high level one is that primary analysis should provide an estimate of the treatment effect that is aligned with the estimate of interest and reliable. Now, I'm not going to talk about estimates and estimate-aligned estimation. The whole day I didn't hear the word estimate. That's fine, but I will focus on reliability for now. What does reliable mean? So how could I characterize a point estimate? What are the desiderata for point estimation? So here's a list of some desiderata. I'm not going through any of these in more detail. I just want to say that statisticians have the list of desiderata. And maybe the first one, unbiasedness, of course, we have talked throughout the day. It would be good to have a point estimate which is unbiased. But this is not really real. We already heard that there's conditional bias, there's unconditional bias, there's mean and median unbiasedness. And many designs, many adaptive designs actually don't provide any unbiased estimates. So then with confidence intervals, I can also have a list of desiderata. Again, lots of topics. But for example, coverage. If I am now in a situation where I'm selecting the best out of 4 populations, it's not sufficient just to take the regular conventional confidence intervals with the 95%, because I have 4 populations, so there's something multidimensional going on. So I need simultaneous confidence intervals, whatever that exactly means. But I would like to have confidence intervals that target not the regular 95% for one objective, but somehow take into account that I have selected the best out of 4 populations. A question comes up whether these confidence intervals should match hypothesis testing. So very often, and we are probably tuned into that, if a p-value is less than 5%, the confidence interval will exclude, for example, the 0. Is that the requirement that I want to maintain? And confidence interval should be informative. I'm not going to explain this, but although I will refer to that on the next slide. And there are other desiderata. And it doesn't matter what exactly it is. The question that I have on the next slide is, are these desiderata universally achievable? And I think I already said it a few slides ago. The answer, unfortunately, is no. And here I have a series of examples of increasing complexity. So the first one is actually not an adaptive design at all. All what I do is to run a clinical trial to compare experimental versus control in an oncology, of course, and where I first test for progression-free survival, and if significant, then I test overall survival. That falls under these fixed sequence, gatekeeping, hierarchical test. It's a very, very standard approach. Whenever I have more than one endpoint, that I test them sequentially. Now, this is protected with respect to type 1 error rate inflation. But unfortunately, there is no methodology available as of today that would provide me informative simultaneous confidence intervals that would respect this test sequence, that incorporate these testing sequences. And such confidence intervals simply don't exist today, not for this type. So what should I do? Well, should I fall back and report the conventional point estimates and confidence intervals because I don't know what to do else? Now, let's make it more complicated and now have the first level of adaptation, which means I add a group sequential design on top of that. So what you see grayed out, so this would be the analysis of PFS and OS at an interim analysis. And if not significant, then I continue to the final analysis, and these are the black dots now, PFS and OS. Now, there are adjusted point estimates and confidence intervals, but only for each endpoint separately, not to take into account the entire testing scheme. Because if on the left side already don't have this information, then in the group sequential design in the middle, which is more complicated, it's even less possible to come up with overall adjusted estimates. Shall I now report conventional point estimates and confidence intervals, knowing that compared to the left situation, I have now an additional level of bias because I might be stopping early when I see overwhelming effects, which can contribute to bias. So I have two sources of bias now. Should I report my conventional point estimates and confidence intervals? Now make it even more complicated because this is an adaptive design workshop. I have to have a real adaptive design. So let's assume we test PFS and OS, but for 2 doses. So I have now 4 hypotheses. I do a group sequential design, that's in the background, and I allow for dose selection at the interim analysis. I have 4 levels of multiplicity. I have 2 doses, I have 2 endpoints, I have a group sequential design for early trial stopping, and I have a dose selection at the interim. What should I report? I really don't have the answer. As a statistician, I'm stuck. And this is why I would like to turn the table. Can we now ask whether there's any sort of ethical guideline that could help us with respect to reporting estimates? So what information is most relevant to healthcare professionals and should therefore be reported at trial end and be included in the product labels such as SMPC or the USPI in the US? Are there ethical considerations that guide us in defining what qualifies as a reliable estimate? And maybe the scrutability, if I pronounce it correctly, maybe goes into that direction. Are there statistical desiderata that I had on the last slide? Are they aligned with such ethical considerations? And if not all these desiderata can be met, which ones should take priority? And is there a risk that in trying to be statistically correct in respect of one desideratum or another, we actually confuse or mislead the prescriber who's reading the label? At this point, I realized when I was putting the slides together, I'm not just a statistician, I'm also a patient. So I ask myself, what do I want to see in a label? Would I prefer an unbiased treatment effect estimate with very high variability, which essentially means that the actual results in the trial are not very reliable, because due to the high variability can be all over the place? Or would I rather have a measure that balances bias and variance in some way, for example, mean squared error, for easier interpretation? If my physician uses the product label as basis for their recommendation, can they trust it gives the full picture, given everything what I just said? Would I, as a patient, still choose this treatment if I knew the treatment effect estimates are based on numbers that even experts, like the ones here in this room, debate how to interpret? So putting all this together, is it acceptable to continue reporting conventional treatment effect estimates and confidence intervals, and only when results are statistically significant? Now, if we agree that conventional estimates are potentially misleading, what would be meaningful and feasible alternatives? How, in particular, should we evaluate complex innovative design proposals where estimation is not straightforward? Or in other words, and I think David White had this question in the morning, I believe at least related, if I have a design proposal which addresses a relevant clinical question, it's foolproof with respect to type 1 error rate and other important considerations. However, the interpretation of the estimates are unclear at best, or maybe even misleading, would the proposal for such a design, could it even be rejected because of that? And how should we balance transparency in communication with methodological rigor? If I continue reporting certain types of estimates, for example, the conventional ones, I know as a statistician there are limitations. How do I communicate these limitations to the outer world, to the customers of this information? So how do we balance transparency in communication, methodological rigor? So I started my talk with Swiss Army knives, and I would like to end with it. Adaptive designs, are they the Swiss Army knife of modern clinical trials? The answer is, as usual in practical life, it depends. If I have a specific task of cutting a piece of paper, what is the optimal tool? It's a scissor. It's the optimal tool for a specific task. There's nothing else that can do it better to cut this piece of paper. This is the regular two-armed RCT without any interim analysis and whatever. Now, of course, sometimes I would like to have some flexibility, and that's where my beloved Swiss Army knife comes in. It's a versatile tool that combines several individual functions in a single unit. Great to have it. I can still cut a piece of paper, and probably reasonably well, so that's good. Now, believe it or not, there's something called giant Swiss Army knife. It's not a fake, it's real. I have seen that in a shop. It's amazing. You see it, you want to have it, right? It's a 2-pound gorilla, 150 to 200 functions. But I also knew it's very expensive. If I would buy it, I would never cut a piece of paper with it. So I leave it at that. I just want to thank, I have been working over the last few years on the ICH E20 expert working group on adaptive design. Hopefully that will come up very soon. And yeah, a few individuals who helped me along the lines along the years in formulating my thoughts. Thank you for your attention.
Great. I may be biased, but I thought that was a really good talk. Questions, Alex?
So thank you. That was really, really great, and it helps me articulate something that is always frustrating when I talk to really smart people like you. And I can put it now as like a problem of transitivity. Should I still want transitivity? We start out with the very simple Swiss Army knife, and then it's like, you know, there's a design that dominates that. There's a very simple adaptive design that's going to stop one patient from having an adverse event. And so surely you should prefer that small adaptive design to the original, I guess this is the scissors, sorry, to the scissors. So a very small first Swiss Army knife is going to be better than the scissors. Thank you. And then it's like, but now I can repeat that. I can also adapt in another way, and I'm going to get another efficiency, and so on until I have the giant thing there where it's like, I can't carry this in my pocket. What can I do? Right? And so is there like a just noticeable difference at some point? Because surely at the beginning, there's something that's better than the scissors. And I presume part of what we're trying to do here is find the sweet spot in the middle.
Yeah, no, thank you. That's great. And I did not meant to say that we should never look into adaptive designs for sure. But yeah, to me, this is actually on that way. And the group sequential design has clearly ethical advantages, and I think we should look into this. Likewise, stopping for fertility, clearly. So whenever we have very clear rationales, we should do that. Now this one, it looks complicated. It's not the most complicated design that I have seen, right? And we have talked about many other complicated designs today, but and they might all have good reasons, but that one is already, to me, is already very complicated. So, and to understand, how do I now report the results? I have no idea here. I already have difficulties in the previous versions, and now imagine the more complicated ones. And now the questions to me is, yeah, where's the sweet spot? And can I reject a design proposal which is really fit for purpose and addresses a good question, but I can't interpret the results? It's not scrutable. Yes. Yeah.
Frank, that was a fantastic talk. Thank you. I just wanted to put out there that you're talking very much about the phase 3, the confirmatory type adaptation.
Yes.
In a phase 2, multi-arm response adaptive randomization, there is an odd contrary effect, which is the response adaptive randomization reduces the bias in the point estimate of the selected arm, not the others, because they've got fewer people up. It reduces type 1 error. I'm sure you can't compute, and there's no way computing a frequentist confidence interval for it. But I just wanted to put it out there that not all adaptive designs result in bias and this selection problem. That actually, in its place, the response adaptive randomization has these very positive properties. It just kind of ironically, you don't use them in the confirmatory setting.
Maybe we can talk it offline because I'm not sure whether they're free of bias. And
I didn't say they were free, but they have less bias than the fixed trial that does the same thing.
I think we need to define exactly how we can compare apples with apples.
Okay.
Very happy to do this. So, yep. Mm-hmm.
Great. Any more questions? Yeah, John.
I mean, to be provocative, I mean, one take one might be I should give up with p-values, especially when I'm using gatekeeper type processes, and I should just focus on estimation. And there'd be a hardcore Bayesian view, which would be your posterior is your posterior. Anyway.
Yes.
I'm not saying I agree with that. I'm just saying it's a view.
No, I understand, but for example, here there's no frequentist or Bayesian on this slide. Nothing. This is a fundamental problem of selection and selective inference. So I'm not sure the Bayesians necessarily can not have that problem.
I mean, yes, I agree, but there is a, you know, on one level, your posterior is your posterior for each of those. Mm-hmm.
Yes, but you're still making a decision, and as soon as you make decision, then you introduce.
You need to allow for that.
Yes. And so I think selective inference problems remain. So, and this to me is an example. It has nothing to do with hypothesis testing. This is just about introducing.
I agree, but that's also an easier one to cope with in many respects. At least read David's papers, but everyone. But yeah. So it's a question of what's the most important thing to focus on, I guess.
Okay, no, that's a fair question. I feel all 5 are important. I don't want to have a drug, and this is number 3, erroneous conclusions. I don't want to have a drug which doesn't work to be marketed. So I want to control the probability of false positive conclusions, but I also want to have interpretable treatment effects. Okay.
I guess this is the thing about, well, you know, 0 is a point on the continuum, and in a way, the estimate of efficacy is more important than the p-value. And certainly, we do this funny thing where we separate registration and reimbursement, for instance.
Yes.
You know, which in some respects is not sensible, but yeah.
And I'm not defending the system. I think all what I'm saying is maybe we should focus more on estimation.